I'm Michael Meshes. I'm the director of the Animal Behavior and In Vivo Neurophysiology Core. But before I start, I want to thank Peter and Vanessa for organizing the seminar series. It's really great that the university is showcasing all of the various cores on campus, because we all have sort of a similar mission of facilitating the production of excellent research. And it's a great opportunity to do that. And I'm looking forward to more such opportunities. I've been the director of the animal, so as the name of the core suggests, Animal Behavior and In Vivo Neurophysiology. This was previously two cores that we recently combined into a single core. I've been the director of the Animal Behavior portion of the core since 2008. And I recently took over directing the In Vivo Neurophysiology portion of the core just in the last year. So if I mix up core and subcores, forgive me, but it's still a relatively new game here on campus. But overall, our mission is to provide the space, equipment, time, and expertise to facilitate research using animal behavior, neurophysiological recordings, stereotaxic guided electrode implantations, as well as injections and infusions into the brain. Though, you know, as Richard said, we're not really with the stereotax. There's other parts of the body that you could address. Sorry, I have my notes covered up here by everybody. Let me see if I can get rid of that. Although I direct and administer the core, none of it would be possible without the outstanding staff who run the core. Nicholas Biscay is an instructor in neurology, has managed the animal behavior core since 2011. He's an expert in all aspects of experimental design, implementation, and analysis of animal behavior tasks. One of the great things about Nicholas is that he comes to animal behavior with an ethological background, whereas I come from more of a traditional physiological psych background. And so between the two of us, we have really sort of a divergent view of how things go in behavior, but it works together very well. Connie joined the core right at the beginning of the COVID-19 shutdown, but she put her vet tech background, her master's in biological sciences, and her experiences at PRA quickly to work to learn how to perform many of our animal behavioral tests. And then earlier this year, she started learning how to do stereotaxic surgery, including both the implanting of electrodes as well as intracranial injections. And of course, she also has been learning how to do EEG recordings in both rats and mice. Jessica has a master's degree in neuroscience, and she actually started in the In Vivo Neurophysiology core, and then took about a decade to work with Dr. Brooks Kayal, learning all aspects of in vivo neurophysiology experiments studying epilepsy models in mice. This is a great group of individuals who really help advance all our work, and I'm very pleased to work with them. This is the Neurotechnology session, or one of them, and we are one of the 5 cores administered by the Neurotechnology Center. You can go, as Richard said, to the Neurotechnology Center website and find out more information about the center as well as each of the cores. The Neurotechnology Center also has produced what we call the core management platform, which I'll show you more about later, that allows you to help do reservations for each of the different facilities. Currently, we are doing all of our experiments in RC1 North, both for the animal behavior and in vivo neurophysiology. We have space in the animal behavior suite, as well as in suites 1 and 3 for mice and rats. But we will be moving to the Anschutz Health Sciences building sometime early in 2022. The rest of our offices are located over here in the School of Pharmacy building on the 3rd floor. Animal behavior permits investigators to really more thoroughly phenotype rodent models of disease and normal function, as well as to test hypotheses of the mechanisms underlying the various behavioral endophenotypes that are related to the disease. Neuroscience research often depends on animal models, mostly in rodents, and being able to objectively quantify variations in behavioral phenotypes in those models is a cornerstone of the neuroscience endeavor. So the animal behavior subcore provides investigators with access to really high quality behavioral testing equipment for mice and rats, as well as our expertise in consultation in all steps, from planning the experiments to publishing. The core provides the facilities to do all levels of behavioral phenotyping, from cognition to emotionality, social ability, motor function, sensory function. These are, in combinations of these, and some aspects of these are important controls for others. For example, if you're looking at, say, a fear conditioning task in which a mild foot shock is administered, it's also important to know whether or not that particular animal model has changed its sensory function. So we integrate all of these different tasks to make sure that really rock solid data is being produced by our users. We can help with the whole gamut of animal behavior, from developing an experimental strategy to either training you how to perform the tests, to working with you to do the test, or as a turnkey solution, we'll perform the tests and provide the data for you. In addition, sometimes, though I'll show you in a minute, we have a whole lot of tasks that we can provide. None of those tasks are quite the right fit for your particular question. And so we are also adept at developing new tasks and bringing them onto campus, validating them, and getting you up and running with that particular task. So one of the things we provide is simply space. If you have all the behavioral neuroscience background you need, but you don't have the place to do the experiment, and we have some PIs like that, we provide space for those PIs. We have currently 4 rooms in RC1 North. We have a water maze room that allows us also we have cameras mounted in the ceiling, so we can do top view tracking of various mazes, from open fields to elevated plus maze or zero maze. We also have a rat room, which is an equivalent, that we actually use both rats and mice in that room just because our mouse work is so strong. We also have a multitask room that can do other things like rotor rod or paired pulse inhibition, fear conditioning, things like that. And we have a whole room dedicated just to mouse operant conditioning, which we put together in the last couple of years and is being used extensively as well. Just in the last couple of years, we've had more requests to bring our equipment elsewhere. So we also provide some off-site testing that can be conducted in either PI's labs or some in the ABSL suites. So the way this works is that PIs rent space and equipment from the core in half-day increments. And we have an online scheduling platform that I will show you momentarily how that works. So the equipment, we have a whole wide variety of tests, from some of the classic mazes, such as Morris Water Maze, which I'll show you later, elevated plus maze or zero maze for anxiety, open field, as well as some newer mazes that are more detailed. For example, Nicholas brought together a test called the puzzle box, which looks at more executive function and is not as detailed as a particular memory test. We also have specialized standalone equipment for looking at such things such as grip strength or acoustic startle, motor function. And all this is made possible in large part because we also have some very powerful video tracking software from Noldus that allows us to analyze data. And I'll show you a little sample of that also later. While our tasks do center on rats and mice, we also provide equipment for other rodents. For example, we've used some of our rat equipment for gerbils, and we've even asked to develop behavioral tests for pigs, which was a bit of a challenge, a little bit beyond my personal level experience. We do provide a wide range of behavioral tests for cognition, emotionality tasks, which are helpful for looking at things such as autism spectrum disorder or ADHD type tasks, social behaviors, which is also important for ASD, and motor and sensory function. I'm not going to read off all these different tasks. I will, but I encourage you to, if you have a question or wish to know if we can help you with any of these sorts of tasks, to contact us for that. So here's an example of a Morris water maze. The Morris water maze is probably one of the most well-known tests of spatial learning and memory that was made famous by Richard Morris back in the 80s. So the task is simply this. You start with a big tank of water. In it is a hidden submerged platform. And a mouse is placed in the platform and asked to learn where this platform is in space. Its only cues of where to find it are distal cues located around the maze. And so the mouse has started over trials at 4 different locations pseudo-randomly. And over time, it has to learn that this submerged platform exists. And over trials, in fact, it will learn to swim directly to the platform. Now, this is evidenced by the fact that over days of trials, you can see that animals get better and reach some sort of asymptotic level. Importantly, if you take this platform out of the maze and allow the animal just to swim around for a period of time, usually about 60 seconds, you can see that the animal will spend more time crossing over that area where the platform was. That's called the target zone, as well as spending more time swimming just in the general vicinity of that maze, and that target rather, and that's shown here. So this is a sort of a gold standard spatial learning task that a lot of people use. We've had a lot of people who come back after a grant has been reviewed and been told they need to do something like this, and they want to implement it. To analyze this, if you do it manually, it is very, very time intensive. You have to actually overlay this with a piece of paper, trace out the path, measure it by hand. I did this work in graduate school. It's a lot of work. But with our video tracking system, we can actually track an animal from its start location to where the platform is. Here you see him swimming around, and this is a pretty well-trained animal, so he finds the platform pretty quickly. The software automatically superimposes the animal's path on a model of the water maze. Using that information, we can look at the animal's behavior over trials. Here's an example of 2 blocks of 5 trials given over a couple days. This shows a heat map of where the animal generally spends its time, which can be very useful, especially for more complex tests. But that's more of a qualitative assessment. Here's a quantitative assessment where we can actually look at how much, what was the length of the path of the animal for those first 5 trials versus on day 1 versus on day 2. And you can see here is a very significant improvement. We can look at the distance the mouse moved. We look at the velocity. We look at the time it took to get from the beginning to the start location to the platform. All these things are important because if, for example, the animal swims slower and their latency decreases relative to controls, you might say, well, it doesn't know where the platform is. But actually, if you looked at the distance it swam, it would be equivalent to the controls. So having these various types of measures all in place is really a critical aspect of interpreting behavioral data. So how do we book a room? Again, the Neurotechnology Center core management platform is available through the Neurotechnology website. But if you want to go to it directly, the URL is ntc-cores.cuanschutz.edu. If you click on that, it'll take you to a login page where you select to use your university credentials. The next page, you can just use your university credentials to log in. Hard to see here, but at this drop-down box, you can select which of the cores you want to use. I helped design this, so animal behavior shows up first. But you could select this and select the ALMC, or the in vivo neurophysiology core, or the one core as well. Select the day you want to book. A little pop-up window will come up. You can fill in what tasks you want, how long you want to do it. Importantly, you can add the PI's name and speed type. Then you'll be billed automatically on a monthly basis. Time may be really one of the most valuable resources that we provide. We can do everything from training you or your personnel to do the experiments, to assisting you in performing the experiments, to just in a turnkey fashion doing the experiments for you and providing the data and as well as the interpretation. But honestly, our preference is really to train people how to do the behavior, because really until you get into the guts of an experiment, it's hard to really understand all the subtleties that are involved with it. But I would say by far our very most important resource is our expertise. Nicholas and I have spent over 50 years in animal behavior, and Nicholas is currently a member of the IACUC, which comes in really handy in helping people navigate the process of actually being able to do animal behavior experiments on campus. We provide guidance with all aspects of animal behavior experiments, really from the beginning to the end. And we strongly encourage anybody interested in performing animal behavior on campus, whether you have the equipment and space yourself or not, to come talk to us, because we can really help in a lot of different ways. As I mentioned earlier, sometime early next year, we'll be hopefully moving to the Anschutz Health Sciences building of new Vivarium. And that really should help alleviate the scheduling issues that we've been experiencing recently. So these new space is going to give us more room, with 5 rooms each for rats and mice. The ability to observe the test from the central control room, which really helps decrease the influence of the observer on the behavior. Right now, the observer is in the room with the animals, and that obviously can have an interaction. We also have specialized, so also the expansion will give us the chance to have specialized rooms for each type of experiment, either whether it's an experiment that requires a top view video tracking, or ability to do water maze. And in our new rooms, we'll actually have hot and cold water, so we'll have better control of the temperature of the water, or operant conditioning, etc. Nicholas and I were really fortunate to be able to help design these rooms. And so one of the things we did was create improved lighting that allows us to do indirect lighting that will help with the top tracking, especially in water, which is prone to glares, and also provides better replication of the lighting environment. So if you're doing an experiment, now or 6 months from now, you'll be able to get the exact same level of lighting much more easily. But really, one of the features that we're most excited about in the new space is that each of the suites will have a temporary housing rack that will allow us to bring the animals in well before the experiments, so that the stress of moving the animals from their vivarium space to the behavior suite will be removed, and we can probably provide more replicable and experiments and easier to perform. So please reach out to me or Nicholas for more information. You can reach us at our email or on these phone numbers, but really the email is always the best bet, because Nicholas is generally busy down in the behavior areas. Video EEG monitoring permits investigators to more thoroughly phenotype the rodents they use in their research, and specifically address issues of whether they have abnormalities of neurophysiological function, such as EEG background suppression, sleep disturbances, and seizures. This can be important for the characterization of translational models of nervous system disorders, including stroke, epilepsy, traumatic brain injury, as well as neurodegenerative, psychiatric, genetic, and developmental disorders. Over the last decade, the in vivo neurophysiology subcore has implanted thousands of rats and mice, recorded hundreds of thousands of hours of video synchronized EEG, performed hundreds of stereotaxic injections, and has contributed to papers and grants and trained students, fellows, and faculty. And honestly, I should have put a similar slide up for the animal behavior core because we've been just as active in all these types of things in the animal behavior core as well. The subcore provides synchronized video and EEG recordings for cortical electrodes, as well as local field potential recordings using depth electrodes in awake and behaving animals. This is really important because a lot of neurophysiology is done in either slice preps or anesthetized animals. We actually do these in awake and behaving animals, which provides more challenges, but it provides an animal model that is probably more realistic to what's actually going on. These recordings can be performed for 24/7 for days or even weeks. And we currently have the ability to record simultaneously from 16 rats and 16 mice. Here you can see our mouse setup with 4 different stations of for 4 animals each. Here's a recording showing a EEG seizure occurring. And in the last year since I took over the core, or as I took over the core, we've also developed the ability to record visual evoked potentials from both surface and depth electrodes. Another big part of what we do is stereotaxic surgery. We implant electrodes as part of one of the major features of what we do, but we have developed we have purchased robotic stereotaxic apparatus that allow us to very accurately place needles for doing infusions and injections in specific parts of the brain. We can do these surgeries for you, or we can train you and you can rent the room, which includes the anesthesia machine and as well as the stereotaxic apparatus. We also provide review stations with licensed clinical EEG analysis software for analyzing seizures, sleep cycle, EEG power analysis, a wide variety of things. This is becoming less important because people have generally more powerful computers on their desktops these days, but we also provide guidance and expertise on how to do those analyses if you're going to do them yourself. For those labs that don't have the capacity to verify the electrode placement, in the last year, we've implemented the ability to do that as well. And also with the addition of Jessica to the core, we've added the ability to do the data analysis for you if necessary. And we're really excited to be able to provide that as well. Similar to the animal behavior subcore, we also provide the training, consultation, and grant support. Importantly, especially when working with awake and behaving animals, is everything you do has to go through the IACUC committee, and we'll provide boilerplate templates for the behavior and in vivo neurophysiology stuff, as well as doing the histology. We provide layers of support for grants. We help with obtaining preliminary data for grants. If the project is complex enough, we'll act as co-investigators on some projects. So we're available to do a very wide variety of things for you. So please feel free to contact me, Connie, or Jessica to access the IBNC subcore services. Again, email is probably our best bet here. So I'd like to acknowledge the tremendous amount of support provided by Mark Delacqua and Paula Robinson in the Neurotechnology Center, which is generously funded by the Dean's Office. Our users, of course, provide funding via charges for services. And really, I'd like to also thank Nicholas Biscay for help with putting this whole presentation together. Overall, our goal is to facilitate the production of really high quality science by providing the expertise, facilities, and equipment required to perform animal behavior and in vivo neurophysiology experiments and intracranial injections. So, let us know how we can help you. We're around, we're accessible. Let's get together and talk. So please let me know if you have any questions at all.
Mike, thanks. It's a great core, great talk. And I just have a question, more like logistical question. We have a lot of users who are applying animal imaging, non-invasive animal imaging, and their mouse models. And obviously some of those are your mice. And I'm just wondering how difficult it would be to you know, to take the animals to the animal imaging, to MRI mostly, to MRI suite and return it back. Do you think it's something we can explore in the future? Because so far, I don't think it has been happening.
You know, that's a great point. And right now, if we're able to do it, now would be the time to be able to do it, since we're both in RC1 North. It is going to be more complicated once we move over to the new building, because that vivarium space is now going to be contiguous with the R2 vivarium space. And so we'll have to work out protocols for doing that. And this is something that we work with people all the time on, because, for example, I've got a PI who wants to do some experiments that involve blast injury over at National Jewish. So organizing how to move animals into a facility is part of what we do, and work with Jory and her staff closely on doing that. But, you know, we should talk more about the order of operations required to take advantage of these two very powerful techniques. In fact, I was just reading a paper about electrode placement verification using MRI in animals, and that would be great if we could be able to do that prior to investing the time in recording from the animals.
And we do this kind of MRI scans for the Jaeger strepper, for example. So
Right.
Let's just talk further. Thank you.
Absolutely.
Yeah. Sorry, Jory Lozinski. Natalie and Mike, that is something we are very aware of, that animals and the ability to move animals between R1, R2 and AHSB is very much on our radar. And we want to make sure that any of those barriers are as minimal as possible. So that is something my group is actively working on, so that, because we fully anticipate those collaborations and those uses of the course. So just for everybody to know, that is something that we are trying to make sure that we set up mechanisms to manage.
Great.
Thank you.
Any other questions? Well, thank you all for your time and attention. I hope this was illuminating as to what the animal behavior and in vivo neurophysiology course can provide to our campus. And I look forward to the next of these seminar series.
Thanks very much, Michael.